Hello, everyone. My name is Avi Srivastava. I am currently a postdoc in Rahul's lab. Today, I'm going to talk about a very exciting new single cell technology called single cell cut-and-tag-pro, and how can we use it to study the functional role of DNA elements through computational tools called single cell ChromHMM. As a computational biologist, I'm very interested in understanding the functional role of DNA elements. And one way to study that is by studying the modification to the tails of histone proteins. And the basic idea stems from the histone core hypothesis, which basically states that the combination of modification on the histone protein correlates with the function of surrounding DNA elements. For example, the presence of H3K4 trimethylation modification indicate a functional promoter, while H3K27 trimethylation indicate a functional repressor. But there is a whole suite of these modification, and the idea is that not one histone protein alone, but the combination of them can help us define the genomic function, which is really exciting from the perspective of a computational scientist. Now, there has been multiple efforts in the past to study the functional role of DNA elements. For example, ChIP-seq, which was developed back in 2007, which is a great for profiling histone profiles in bulk, but it doesn't really scale well to thousands of single cells. And then single cell ETAC-seq which was developed relatively recently. It can profile open chromatin region at single cell resolution, but we can't really learn the full regulatory landscape with open chromatin profiles alone. For example, poised and active region we cannot differentiate between them using open chromatin profile alone. So in advance to that, there was a single cell cut and tag, which can profile individual histone modification across thousands of single cell. But the data from this technology is relatively sparse. But more importantly, not only sparsity, we can only measure a few histone modification within a single cell. Note here, to define chromatin state or learn the regulatory landscape, we have to profile multiple histone modification within single cell. So within the same single cell, sorry. So to improve on that, we develop a technology called single cell cut-and-tag-pro, which can profile both histone modification profile as well as cell surface protein within a single cell. And the basic idea is to stain the cells with antibody targeting both cell surface protein and histone protein, and basically run 10x single cell cut-and-tag profile protocol. Using this method, we co-assay some 134 protein along with on 600 histone modification profile, where 4 of them were active histone modification and 2 of them were repressive histone modifications. So as a listener, the first question that might come to mind is, why profile protein at all? Single cell cut and tag is there, why can't we use that, right? So why single cell cut and tag pro, the protein part? The short answer is sparsity and the cell type specificity. So here I'm showing the unsupervised UMAP clustering of the H3K4 monomethylation dataset, where the cell type resolution of the single cell cut and tag data alone is not very high, while protein-based clustering in the middle shows very high cell type resolution, which is very useful for sparse data so that we can group similar cells together and boost the histone modification signal compared to the background. As we show on the right as well, the weighted nearest neighbor analysis, which use the information from both the technologies and define a combined clustering. And as you can see, there are multiple cell type annotation which we can learn from this data set alone. So to make sense of all of this and learn the functional role of DNA elements at the single cell resolution, we extend the previously developed method of ChromHMM into single cell ChromHMM. Through the next couple of slides, I'm going to go through this process step by step. So if you think about the overall goal of studying the functional role of DNA elements, it's pretty broad. To simulate that, I'm showing on the right is a black box, which takes in single cell profile of 6 histone modification and outputs the functional role of each DNA element. And throughout the talk, we are going to unwrap this black box by solving multiple smaller subproblems for understanding the basically functional role of DNA elements. And if you think about the first subproblem, the first thing which we have to solve, since each of this histone modification experiment is individual, it's very independent. Since we cannot profile all of them within same cell, we need to define a common framework to compare these different experiments so that they are on the same coordinate, and we can define their cell type annotation or learn their chromatin states. And there are basically two ways we can assume defining these systems. First is supervised way, where we assume one protein as the reference and transfer all other modalities onto that single reference. But the problem is that it is limited by cell time resolution of the base modality, which was chosen as the central modality. Another way is to use independent data set where we know very high cell type resolution, and then we transfer everything onto that. The advantage there is if the deeper sequence data is already present, then we have information which gets propagated down to the newer experiments. So that's what we did. We previously designed a reference framework which uses protein or RNA. It's a very dynamic framework, but here we are using is a protein as the central modality and anchor to integrate data from all the histone modification profiles which we profile in this experiment. As shown in the left, we have site-seq as a reference, which is very deeply sequenced data set with high cell type annotation, and integrate single cell cut and tag profile along with the ATAC-seq along site-seq already have RNA-seq data set onto it. And the basic idea is, as shown on the right, is that it will enable joint visualization of so many different molecular or cellular modalities for downstream analysis and comparison. So the best part of our data is that we have 9 cellular modalities that are each measured in a consistently defined set of individual cell types at the top through technologies like ASAP-seq, which can profile ATAC-seq and protein dataset, site-seq, which can profile RNA-seq and the protein dataset, and the newly developed single cell CUT&Tag-pro, which can profile histone modification and the protein. Notice protein is common in all these technologies, which we used as an anchor to integrate everything together. I'm showing you as a positive control the CD8 loci. We have lots of cut-and-tag experiment which were showing patterns of activation across CD8 T cells, while repression across all other cell types based on K27 trimethyl enrichment. On top of that, we see that how these things feed through the other molecular modalities like CD8 loci shows RNA-seq high-level expression. Also, CD8 protein show high level, and it basically serves as a very good positive control example. So this idea of integration unwraps the black box a little bit, and it brings us closer to the understanding the functional role of daily element, right? So basically, to summarize, we integrate single cell profile of 6 histone protein across, let's say, 20,000 cells based on how many cells were in the site-seq reference, and we seek to define a model to simulate the histone code hypothesis, which we talked very early in the starting of the slides, right? So basically what we want to learn is the hidden chromatin states, and the probability of observing them across 200 base pair region across the genome. And chroMHMM is just the tool to do that, which was basically takes the suitable profile of cell types and train a multivariate hidden Markov model by running Baum-Wunsch algorithm on that. It also defines the probability of observing each chromatin state across each and every 200 base pair region across the genome using forward-backward algorithm. And that's what we did. We used bulk chroMHMM on a data set with the suitable profile of the site-seq defined cell type annotation. Very broad cell type annotation we used here. And we learned 12 chromatin states as is shown on the left, broadly defining promoter regions through state 1 and 2 because of the enrichment of K4 trimethyl and dimethylation, 3-4 state defining the enhancer state-like pattern because of the enrichment of K4 monomethylation. Similarly, K27 trimethyl enrichment in the state 9 and 10, and K9 trimethylation for heterogeneity region in state 12. And we use these states to show another positive control example, which is PAX5 on the right. PAX5 is a marker of B cells. As we can see, we show higher enrichment on the B cells for K27 acetyl, open chromatin, and all the mono and ditrimethylation data set. While CD8T and monocytes are showing K27 trimethyl, which is a repressive mark, enriched on their cell type annotation. Along with that, the beauty of this is we can learn the chromatin states right? So here each color denote the chromatin states as defined on the left, where green is marking or marking as the promoter region, blue is marking the enhancer region, while red marks the repressed region across the genome. This is what we want to know, the combination of histone marks which are defining the chromatin state. So this is beautiful. But we wanted to push the boundaries a bit further. So we can do a pseudobulk analysis, but it's kind of unsatisfying both intellectually and practically. So for one thing, it's not great when the level of cell type resolution is not known, right? Here we know, but what if we don't? Moreover, pseudobulking discrete clustering is difficult, if not impossible, when analyzing like developmental trajectories. So we really want to avoid grouping cells into predefined bins, which is necessary for bulk ChromHMM, right? Ideally, we like to keep everything on the single cell level. It would be amazing if we could run ChromHMM on each and individual cell, but this is challenging, especially because the data in each cell is sparse. Moreover, the computational and storage requirement can increase by magnitude because we basically have 50 cell type, but here we're talking about 20,000 cells, right? So to tackle that problem, specifically, we designed a single cell ChromHMM, which is a Rust program to define which perform the chromatin state analysis using the integrated single cell profile. We basically rerun the forward backward algorithm using the HMM parameter, which we learn on the bulk profile through bulk ChromHMM. But the biggest advantage of single cell ChromHMM is that it utilizes high performance computing by performing quantification on highly parallel fashion across each cell, and uses a binary format to store the post-shear probability in a compressed fashion, which saves a lot and makes the problem computationally tractable. Feel free to really read a paper. We put every detail in there. Now, if we go back to a previous positive control example of PAX5, where we are showing that bulk chromatin states near to the transcript start site. But the beauty of single cell ChromHMM is we can go at the single cell level. So here in the UMAP down below the bulk chromatin state analysis, I'm showing for each 200 base pair region. Here, the yellow band is showing the which specific region I'm showing. It is showing highly promoter-like specificity, right? So that's what I'm showing on the UMAP. Each cell with their score on the B cells and monocyte and the CD8 cell, and we see that B cells are showing higher enrichment for promoter-like state. And similarly, in the middle, we see all other cell types shows repressive state-like pattern, and similarly, PAX5 gene expression is higher in the B cells as a positive control example. So it's again showing at what resolution we can go and analyze to see the chromatin states of each and every 200 base pair region for each and every cell. That's very important part. Now, what can we do with that? We observe that chromatin state dynamics learned by single cell ChromHMM for individual cell are highly cell type specific, right? Especially for the repressive state, I'm showing on the left, where we kind of look at the TSS transcript start site probabilities. Not only that, the learned chromatin dynamics enable us to explore how genomic function varies across a trajectory. Here I'm showing the CD8 T cells trajectory, how it goes from naive cell, from T memory cell to effector memory cell. And lastly, on the right, I'm showing an example of TBX21 and LEF1, a transcription factor that's known to be a key regulator of T cell maturation and differentiation. And as we move along this pseudotime trajectory, we can see that for cells that early in the pseudotime peaks continue, early in the pseudotime trajectory, we can see that for cells that early peaks contained in these motifs are open or closing. But as cells differentiate, they either increase or start to decrease based on what cell states they are in. So lastly, in summary, to summarize everything we have talked about, we developed a new technology to co-assay histone profile and cell surface protein within an experiment. It empowers us to enable the computational tool which explore the functional role of DNA element at single cell resolution. Another thing which I couldn't talk today, and it's very exciting due to the time limit, is we can define a mega omic atlas of the PBMC cell to transfer profile across new experiment where we don't have simultaneous protein measurements. With this, I'd like to acknowledge all the members of the Satija lab for providing valuable feedback throughout the progress of this project. I also like to thank all the innovation lab members and all our funding agencies. So if you're interested, please feel free to reach out, read our paper online, or follow us on Twitter. And with this, I'd like to thank you all for listening, and I'm open to questions.